rest of the issue, there were some other things that should be fun to talk about. And a couple of them here, I'll just do one. They're related to each other. Has to do with the periodic table of the elements in chemistry. And this is a brief little news item uh, titled "Covering the Elements," and it starts off like this. Should be fun stuff here. Looking for some reading inspiration this summer as you ride out the COVID-19 pandemic at home? Look no further than Nick Thomas's Periodic Table of Element books. Thomas, a chemistry professor at Auburn, has assembled a periodic table in which each element is represented by a book containing the element's name and its title. Many of the books Thomas has included are not about science. For example, there's a collection of poems called Iridium, a business book called The Francium Rule, and a book called Mercury that's not about the element, but about the late British rock star, Freddie Mercury. And by the way, I mean, many of you are probably familiar with the story of Freddie Mercury, but if not, that is a truly inspiring life history that you might want to take a look at sometime. One criterion Thomas uses for inclusion is that the book cover needs to be eye-catching so that his completed table would appeal to a broad audience. According to him, academic books tend to have a very boring covers, which probably is generally true. Thomas tells Newscripts that the idea to assemble a periodic table of books came to him when he was still a teenager. While other kids collected coins, stamps, and baseball cards, Thomas collected chemistry books. I remember thinking, wouldn't it be fun to get a book that had each element in its title and arrange them in a giant periodic table on the floor? He finally completed his childhood dream this last spring. It turned out to be a fun way to combine some chemistry and literature that most anyone can appreciate even without knowing too much about the periodic table or anything about chemistry. He reveals that one book cover does not contain the name of the element, so we'd have to get a closer view of the periodic table to try to see which one that is. So you can view his periodic table of elements at the following web address: Get Nick T Organic PT Books. Here we go. getnict.org/PT-books. So we'll take a look at that. Just looking at a little tiny picture of it here, it looks pretty interesting. So I thought that might be kind of a lighthearted way to start our week back in pharmacy school. I hope everyone had a wonderful Labor Day holiday that was safe and fruitful, and that you come back feeling rejuvenated a little bit and ready to go for another busy week of pharmacy. Anybody come up with any questions?
I just had a question about our exam next week. Since the class is technically all online, are we going to be split into groups for the exam so that we're not all in the same room, or how is that going to work? Or do you know?
My understanding of that, it's going to be conducted like a regular exam. In other words, ink and paper. But I do believe we'll have to divide up. I think the majority of the group will be in 169, but there's a couple of other rooms, and it may be 114 and 117. I'd have to check that to be sure, in which everyone else will be located to achieve the proper distancing that's required by the viral circumstances that we're under. So that's a really good question, and we'll get more information on that as we get going. But assume that it's going to be pretty much the regular format, 50 multiple choice questions, and I'll tell you later in the week exactly where we'll cut that off. At 50 multiple choice questions on paper, as we've grown accustomed to, and the only difference being that because of the distancing requirements, we'll need to split the group up a little bit. That
Yeah,
help
thank
out?
you.
You're welcome. We'll certainly find out more about that as time goes on, and and by the end of this week, have the exact cutoff for inclusion of questions for situation as we proceed through the beginning part of next week and head for late Thursday afternoon next week. 17th, I believe, is the date for the exam. Okay, well, let me try to share a screen with you here. Okay, well, as you all know, we're concluding our introductory segment of the pharmacology course with some brief look at important pharmacodynamic principles and ideas. And as we transition out of this later today, we'll transition into our first drug group, which is the autonomic drugs, which is a historically important group of drugs partitioned along the lines of the anatomy of and physiology of the autonomic nervous system. In other words, the sympathetic and parasympathetic subdivisions of the autonomic system. So that's where we'll start when we get going on the drug classes themselves. In the meantime, a few more segments on pharmacodynamic principles, which are potentially important for any drug group. And some of our fun-loving animal friends here. The last few nights, once again, the raptors have really been out at sunset, and as we're walking the dog, owls and hawks and so on abundant. So it's been pretty fantastic. Hipshot is not interested in gaining the attention of a raptor, though. I'll tell you that. He's trying to stay warm today. And our friend Bob. Okay, so one of the main things we did is that we looked at some graphical representations of dose responsiveness, and of all the pharmacodynamic, principles that are glue that holds our thoughts together about pharmacology. None is more central in my mind than dose response. So it's perhaps not surprising that pharmacologists over about the last 100 years, 125 years, have garnered a number of meaningful ways to represent dose response relationships for a variety of drug substances. And so these very non-specific sort of graphs that we see here, not representing any particular drug substance, but could be any drug substance really that obeys classical pharmacodynamic principles, which is most of them, not all of them, but most of them, would be fodder for representation, as we saw in this series of slides. The idea of risk-benefit analysis is an incredibly important subdivision of pharmacodynamics and dose response, because for whatever drug substance we're talking about, or drug class for that matter, we're hoping for a situation in which we can have maximal efficacy and minimal adversity. And the therapeutic index, as listed in this slide here, which is entitled From Bench to Bedside, which is population-based dose response stuff, looks at the ratio of the LD50 to the ED50. And of course, the larger that number, the better, which basically means there's a substantial spread between the lethal doses and the effective doses. And the therapeutic index, as represented by the LD50 divided by the ED50, is perhaps the most classic way of representing therapeutic index, but it's by no means the only way. And any representation of an adverse effect in the numerator versus an effect, a beneficial effect in the denominator, giving you a ratio between those um, is a potential way of expressing risk-benefit analysis using actual mathematical quantitative formulations. And in the example that's given in this slide, where the therapeutic index is 400 divided by 100, giving us the value of 4, it is quite honestly of a very undesirable therapeutic index because there's not a major spread between the adverse and the effective doses. Typically one would hope for a much greater spread than that to be able to reasonably achieve a particular drug substance. So as we moved into this particular aspect of the slides, we moved from sort of individualistic dose response, in other words, dose response at the level of an individual person, a laboratory animal, or representation in terms of individual test tubes, for example, in which dose responsiveness is being analyzed. We moved from that into the dose responsiveness of populations. And in the dose responsiveness of populations, very often you're looking at the middle of those statistical representations. In other words, the 50th percentile or the median, either effective doses or adverse doses, depending upon what particular parameters you are measuring. So in the development of pharmacology to this point, we're very much dependent upon such population-based estimates of risk and compared to benefit. In the era that we're now approaching, in which the life that you folks will be living as pharmaceutical scientists over the next many decades, where we can better tailor things to a particular individual person as opposed to being dependent upon the averages of the population, we'll probably see a diminished evidence-based dose response and greater representation of dose response for a particular, relevant to a particular individual, which is where we want to be. Because if we're looking at the median of that population and the individual finds themselves either on either sort of far-flung edge of that population, the median can be very misrepresentative of what's. So we talked about some various things beyond safety that can be analyzed by dose responsive graphical relationships. And of course, buried in that is, and we've been talking about efficacy here or effectiveness or potency, which technically refers to how much drug it takes to pursue a given effect, whether that effect is large or small, efficacious or not. So as we, let me just backtrack here slightly to remembering that the x-axis is typically concentration or dose, and movement of dose-response relationships left and right across that x-axis are representations of potency. Movements on the y-axis, which is your response axis in some type of response representation. Movements up or down on the y-axis are representations of how effective that drug is, or in other words, efficacy. So being able to distinguish between efficacy and potency is an important sort of matter in the overall picture for dose response. So I think we were right about here as we ended. the other day, and pictured as in synapses uh, with respect to the dopamine will be the subject of many of our analyses as we proceed throughout the year. Um, so we put it up here as sort of a prototype for us to think about as we're moving now into the realm of cellular and molecular pharmacology. Uh, Certainly related uh, biogenic epinephrine and norepinephrine will also be major players for us. And, uh, and all of these signal to us the idea that mechanisms of action for various pharmaceutical substances are frequently, if not always, represented by perturbations that are occurring in the physiological realm with well-known uh, neurotransmitter and hormonal systems, such as we see here again in the neurotransmitter realm with dopamine. So exogenous drug substances, xenobiotics, if you will, are frequently either mimicking what's going on in an endogenous system or blocking actions that are occurring in that system. So, that is not no surprise to you folks, I'm sure, uh, but is hopefully a clear representation of why the preliminary studies that we've all done with respect to biochemistry and physiology are so critical, because the pharmacology is just taking that physiology a step further, basically new substances to the system, either agonists in the cases of where we're looking at sort of classical efficacy, or antagonists in cases in which we're blocking that efficacy. So this the specifics of this particular cartoon neuron are not critical to us at the moment, but rather representations of important phenomena of the physiological realm, such as the synthesis of neurotransmitters, their transport throughout neurons, their signal release from presynaptic sites to typically their crossing the synapse and producing actions at endogenous receptor substances. To be looking at situations also on the presynaptic side as well as the postsynaptic side. So all of those sort of little areas that we just talked about there in the last minute or so are frequently important in terms of identifying sites of action for drug substances of interest to us. So as you well know, there are many different types of physiological receptors which are critical in terms of our understanding of drug action. One of the most dramatically representative of these groups are the so-called G protein coupled receptors, or GCPRs. And the discovery of the relevance of these receptors beginning in about the 1970s, although many of their representatives were known well before that, they weren't known to be included in the superclass of receptors until about the 1970s. And understanding of what's going on with respect to the G protein coupled receptors is an ongoing matter to this day. And literally thousands, if not more, representatives of these receptors are known, many of which are relevant to pharmacology, uh, but some of which may ultimately be relevant, but we haven't really discovered that connection yet, are going to be sorts of things for you to keep in touch with over the years as the research continues to blossom in these particular areas. Notice under a bullet point underneath G protein coupled receptors is the idea of second messengers. And the first so-called second messenger to be identified with cyclic AMP, which actually precedes identification of the G protein coupled receptors. Because I think the first meaningful work with cyclic AMP was done in the 1960s. So, at the time, if some theoretical receptor was being stimulated and that created a signaling change within the cells, that second signaling change was called the second messenger item. And now the terminology second messenger is really a bit confusing, because things like cyclic AMP aren't really the second message, but they're the third or higher order message following activation of a appropriate receptor. But as we already know, once terminology gets entrenched in a particular scientific area, it's hard to dislodge that terminology. So the second messenger concept is one that remains. And I might just suggest that we alter the terminology just a little bit and call it, multiple messengers. And that way we're not getting stuck on whether it's really second or third or fourth along the line. So I'll say a few additional things, most of which you're probably already familiar with respect to the G protein coupled receptors here in a few minutes. But in the meantime, just recognize that there are other receptors beyond the G protein coupled receptors, which are so critical to our understanding of pharmacodynamics, including another huge family of receptors, which are basically ion channels, whether they be voltage or ligand gated. You'll be hearing a lot about these, especially from Katie, but from many of us, because of their predominance, and from a sheer number of receptors, there may not be quite as many as the G protein coupled receptors, but an equally important subcategory of receptors, nevertheless. So there are other types of transmembrane receptors also. So the G protein coupled receptors would be the most obvious transmembrane receptors. But for example, kinase receptors, which are important, for example, in the actions of hormones such as insulin, or the cytokine receptors, which are critical in terms of hormonal substances such as growth hormone, as a good example, are additional types of transmembrane receptors beyond the G protein coupled receptors. So nature has found it very, very convenient and successful to utilize the transmembrane concept for a variety of receptors. And you see these substances very, very well conserved across the evolutionary span because of the vitality of that particular theme. But some receptors, of course, are not transmembrane receptors. And a really good example of that would be the nuclear receptors, of which the steroid hormone receptors are probably the clearest subclass of signaling molecules in that particular realm. So the nuclear receptors is another superfamily of receptors in containing themes which are vital across many different drug groups. And if you recall, one of the main characteristics of nuclear receptors is that they're located in intracellular, sometimes even intraneuronal nuclear representations, as opposed to being membrane represented. So the physiological substances and drugs which are capable of interacting with nuclear receptors are typically very lipid-soluble substances which can penetrate the cell membrane and find the target in the cytoplasm and or in the nucleus itself. And then, of course, there are other types of signaling molecules, of which intracellular enzymes would be examples, which we may not classically define as receptors, but which are nevertheless exerting physiological and pharmacological effects. So there's a huge number of substances macromolecular substances that fit into this realm of receptors or other targets. And more than we'll talk about in the introduction this afternoon for sure. But we'll be introduced to some more of them as we go along throughout the year. And then even then, not have talked about all the different types of signaling substances that exist, but we'll have talked about many of the categories of those as we approach the end of our studies later in the year. So let's just do a few reminder things with respect to some of these subclasses of receptors. Not worrying at the moment uh, about too many specifics, although I'll give some specific examples as we go along. And the number that's quoted here for the number of genes representing substances in the G protein coupled receptors, the number is listed as 800, but I would propose to you that's a vast underestimate. And there are actually ultimately going to be identified thousands of members of this highly successful category of signaling substances. Many of them are involved in sensory perception. A great example would be rhodopsin. Rhodopsin is the most heavily studied G protein coupled receptors and was undergoing significant study long before the actual class of receptors was identified. So there's decades of work representing scientific understanding of rhodopsin. And in fact, one of the subclasses of G protein coupled receptors is called the rhodopsin class of G protein coupled receptors. And indeed, now many of the G protein coupled receptors most familiar to us, such as the dopamine receptors and the norepinephrine receptors, the epinephrine receptors, and so on, are members of the rhodopsin subclass of G protein coupled receptors. So it's a very highly studied category of receptors, even beyond rhodopsin itself. But for many years, the only crystal structure we had in this area was represented by rhodopsin. And only in about the last decade to 15 years have we seen a significant number of other crystal structures available for other, neurotransmitter substances such as norepinephrine and serotonin as an example. So sometimes this category of receptors are identified as the 7 transmembrane domain receptors because, again, as you probably recall, there are 7 segments that wrap like a snake through the membrane for these receptors, and therein being the 7 transmembrane domain idea. The transmembrane domains themselves are highly conserved. They're the most highly conserved from an evolutionary standpoint of these receptors because of the tremendous success that's been achieved by nature by using this particular motif. So sometimes these names are sort of loaded together, and you'll hear a description of these receptors as being the 7 transmembrane domain G protein coupled receptor types of receptors. And so those ideas are kind of linked to each other. Some of the, but not by any means, all of the areas that are highly represented by these receptors are, of course, neurotransmission, as we've been talking about, smooth muscle control, which is so critical in the realm of autonomic pharmacology that we're about to embark upon, that you'll hear a lot more about that in upcoming days. One of our great themes is metabolism. So the redundantly regulated aspects of metabolism are highly represented by the G protein coupled receptors, but of course also by other realms, such as other transmembrane receptors, or even in the nuclear receptor realm. Metabolism is such a critical theme for cellular physiology and pharmacology that really many different models of receptor signaling are represented by metabolic consequences. Cardiac or beyond cardiac muscle, even into the realm of skeletal muscle, rate and force of contraction, is another area. And in another area which is so critical as far as autonomic pharmacology is concerned, in the area of glandular secretion. So these are just some ideas for subrepresentations within the G protein subclass of receptors itself. Here's just sort of a familiar looking cartoon for a generic G protein coupled receptor. There have been many ways that have been utilized over the years to try to adequately represent the three-dimensional characteristics of these receptors in 2 dimensions, which is not easily done, but this type of cartooning that you see here, and you can count the 7 transmembrane segments quite easily from this type of cartoon. The typical way of expressing it is with the N-terminus of the receptor being extracellular and the C-terminus being intracellular. Although, again, there's nothing sacred about that, and some representations occur the other way around. So you have to be a little bit cautious about how you're looking at it with respect to inside and outside of the cell. But cartoons like this don't really give us any feeling for sort of the three-dimensional ability of a ligand substance, physiological or pharmacological, to bind the receptor. And so a cartoon like this gives us a better feeling for that. And so this, what I consider to be very nice cartoon, which was introduced by a famous membrane pharmacologist named Henry Bourne, is actually found in our Katzung textbook, because Bourne is a faculty member in the same department that Katzung and Trevor are located in. So I think you can see the 7 segments represent what kind of looks like a cylinder in this representation. That's a little bit oversimplified, and it's not quite cylindrical, but to think about it in a cylindrical way is certainly fruitful nevertheless. As you can see, this cartoon has an agonist abbreviated once you get inside to the binding site itself as AG. That's not silver, but agonist that's being represented here. You notice that although these receptors are frequently described as surface-bound receptors, I mean, they are in a sense, but the binding site itself is typically a little bit, about a third of the way into the membrane, and associated with various residues of the 7 transmembrane segments, which are producing a binding phenomenon, and ultimately are involved in transducing that message to the inside of the cell via changes in orientation of the 7 transmembrane segments, which of course is difficult to see in a simple cartoon. But the most important thing to recognize, of course, is that the receptor itself is coupled to the G protein, or the GTP binding protein inside the cell. And there are residues on the 7 transmembrane segments that are located inside the cell, which are coupled to the G protein, in which the message is transmitted from drug to binding site and ultimately to G protein inside the cell. So this slide just summarizes some of the many substances which are important endogenous ligands, which are known to exert some or all of their actions via G protein coupled receptors. And that's just really a little bit of extension of the slide we saw a couple of slides ago. Some other things that weren't on that slide, but which were sort of buried in the content of it, would be, for example, inhibitory neurotransmitters such as GABA, acetylcholine, which is so important in smooth muscle and gland function. And by the way, making note that when you look at something like acetylcholine, some of you may be going, wait a minute, I thought that some of the acetylcholine receptors were ion channel receptors. And you'd be exactly right. Some of these categories of substances are represented by more than one subclass of receptors. So even though acetylcholine has G protein coupled receptors of the muscarinic type, it also has ion channel receptors of the nicotinic type. which are very different molecular themes when you get down to it. But it's been productive to have a plethora of binding sites for a number of these transmitter and hormonal substances. And so the first represented substances on this list are relatively small molecules, but then when we get to peptides, of course, we're talking about much larger substances. And indeed, the endorphins would be an example of that also. And so it's not just small things that are represented by binding to G protein coupled receptors. The small things pretty much are associated with the rhodopsin subclass of receptors, and many of the larger substances that bind to G protein coupled receptors are in the secretin subclass of receptors, which are generally much larger ligands. Um, there are more than two subclasses of G protein coupled receptors, though, beyond rhodopsin and secretin. Um, but most that we know of so far, receptors are members of either the rhodopsin or the secretin subclass of ligands. So, relatively recent additions to these categories would be binding sites for the endorphins, which were recognized by about the 70s, late 70s or 80s. And then even after that, the cannabinoids, which are so important in terms of physiological and ultimately in terms of pharmacological function. So these are just further examples. So the G proteins are found in multiple subforms themselves, and we don't need to spend a long time on this right now, other than to recognize and remember that this movement of information from drug or neurotransmitter to receptor to G protein is headed towards another category of substances known as the effectors. And so in the case of cyclic AMP, the effector would be adenylate cyclase, which is the enzyme responsible for converting ATP to cyclic AMP. So in this particular sense, the G protein itself is kind of like the second messenger. Got the original message from ligand to receptor, second message in terms of that representation with the G protein, and then on to the effector itself, which would be the third level, and cyclic AMP, of course, appears at that particular level. A huge increase in understanding of these receptive substances came with the technical capability of doing molecular cloning. And no surprise that sort of the ability to do that in the 70s and 80s was coupled with a huge explosion of understanding about the G protein coupled receptors themselves. And there are many of these receptors which have now been identified in terms of molecular cloning, in which we still haven't really been able to tie that together with a discrete cellular ligand. So sometimes the additional receptors are known as orphans. And along the way, sure, in terms of your familiarity with these receptors over the next many decades, many of these orphans will find a home. And so you'll be able to reflect back on this moment in 2020 and say, wow, we certainly know a lot more about these receptors now than we did back then. So G proteins are trimeric. They are represented by alpha, beta, and gamma subclasses. And then within those areas, there are subtypes. So there's alpha 1 and alpha 2, for example, beta 1, beta 2, and beta 3, which complicates studying, but greatly facilitates the physiology of these substances. So one of the things that we'll do as we march into the pharmacology associated with some of these areas is to identify particular drug substances which are relatively selective in terms of binding to various subclasses of receptors which are linked to these subtypes of G proteins. So the major business end of the G proteins are the alpha subunits of the trimer. And there's, considerable homology amongst these different subtypes here also, but not as great as the homology that we find with respect to the transmembrane segments. And in some cases, there's very little homology amongst different G proteins. And we'll try to identify some of those areas as we go along into various drug groups. So with respect to G alpha, the GS subclass or stimulatory subclass of the G alphas, one very well known example would be in the case of adenylate cyclase and cyclic AMP, as we've already talked about. But you can also see subclasses in which, rather than driving the concentration of cyclic AMP up, the cyclic AMP concentration is driven down. And this would be, for example, the GI or GO subclasses, GI standing for inhibitory G proteins, in which you see decreased cyclic AMP formation rather than increased. And then the GQ subfamily, which doesn't have anything to do directly with cyclic AMP itself, but has to do with membrane lipids such as phospholipase C and IP3 and diacylglycerol. So there's a lot of different molecular themes represented by the G proteins themselves, and those will play out interestingly, I think, for us as we move through specific examples of these substances later in the semester. So we can do a quick look at some key characteristics of ion channels and some of the other types of receptors, which will take us just a few minutes as we start in our morning session tomorrow, and then on into later in the morning tomorrow, on into autonomic drugs. So tomorrow's our double day again, 11 o'clock and 1. And so I'll take any questions that people might have now. And if not, see you all tomorrow morning.